Hello, and welcome to Informed in 30, a new series from the National Brain Tumor Society designed to give you, our patients and caregivers, everything you need to know about cutting-edge advancements in brain tumor research and treatment. Our topic today is innovations in pediatric brain tumors. My name is Rachael Kittleson, and I'm the Director of Community Relations at the National Brain Tumor Society, and I oversee our Connections program, which provides support and assistance to individual patients while advancing connection and care for the entire brain tumor community. I am also a member of the brain tumor community as a former caregiver to my daughter, who passed away from glioblastoma in 2017. Joining us today is Dr. Kirk Tanner, National Brain Tumor Society's Chief Scientific Officer, and Dr. Mariella Filbin, Dana-Farber and Boston Children's Cancer and Blood Disorder Center's co-director for Research in Pediatric Neuro-Oncology program. Thank you both for joining us today.
Oh, thank you, Rachel. I'm so happy to be a part of this Informed in 30 series. It's really important, and it brings really cutting edge and timely information to our brain tumor community. And we're particularly fortunate today to be really joined with one of the true leaders in pediatric brain tumors, Dr. Mariella Filbin, a physician scientist who's really at the forefront of research, translation, and really importantly, patient care. So welcome, Dr. Filbin. Thank you very much for having me. It's a pleasure. So without further delay, why don't we get started? So let's start out with an overview of pediatric brain tumors and what that means to our brain tumor patients. Mariella?
So thank you again for having me today. I'm really delighted to be on with you guys to tell you a little bit about the new perspectives and also hopefully giving you new hope for this field of pediatric brain tumors. As you all know, pediatric brain tumors and other CNS tumors are the second most common cancer of childhood. However, since the mid-80s, they are the leading cause of cancer-related death in children. That's shown over here in the middle, and not only is it tragic that brain tumors are the most common cause, also our progress has not been as steep and as hopeful as in other cancers like in leukemia. Why that is the case, there are multiple reasons for that, and I've written down some of the challenges on the side. It used to be super rare that we got tissues out of the operating theater. We didn't have many faithful models that would reflect what we see in patients. Our previous and older classifications were oftentimes based on adults. And last but not least, brain tumors are located in the most complex and least understood part of our body, the brain. But there's also plenty of reason for hope. And in my talk today, I want to show you some of the latest advances and how this field is moving quickly, and hopefully will soon be at the point where we have more impact for our patients. So first, I will touch upon new classifications. First, we really have gotten better in the last few years of giving the tumors we're treating a name. You can see here genome-wide DNA methylation profiling as one of the new tools we're using for our integrated molecular diagnosis. On this graph here, each little dot represents one tumor, and over 2,800 tumors are shown here. And they're clustered by similarity, and gave us a first and really detailed view on how many different brain tumors there are. Currently, there are more than 82 different classes described, but even within the classes, there are subtypes. And this is just one way to classify new tumors. Another way is by DNA mutations. And I'm just showing you one of the biggest discoveries of the last few years also, which was the finding that high-grade gliomas of the midline, the thalamus, the pons, and the spinal cord, all or almost all have the same exact mutation in the histone, are called histone 3 K27M mutations. These findings, almost a decade old now, have really transformed this field, have allowed so much more research, and most of all, have allowed for a new classification that is the basis for everything we do. And that's also in summary for this first part of classification. This is the first step of bringing us closer to personalized treatments and understanding more about the biology. Second of all, I want to tell you a little bit about a new appreciation we have for heterogeneity of these tumors. And I'm going to show you a small video we've prepared in the lab. We used to think that a brain tumor occurring in the brain is a tennis ball or a lemon in the brain, and if you had cut it open, you would see all the same kind of cells. But with our latest technologies in hand, we were really surprised to see that these brain tumors are comprised of a multitude of different cells, not only tumor cells shown here in shades of purple, but also multiple normal cells in all the other colors. We've learned this by putting these tumors into tubes and making single cell suspensions out of these, then sorting these cells into wells, and then sequencing every single cell within those brain tumors called single cell RNA sequencing. What we can do with that information then in the end is classify each cell within the tumor. So not just the tumor as a whole, but each cell, the normal cells and the tumor cells. And I'm showing you one example for what we've learned from that. For example, in these histone mutant gliomas, we've learned that tumor cells are very distinct from each other and follow an architecture or hierarchy. On top are stem cell-like cells that are the ones that are dividing and really drive the tumor. But we also learned that about a third of the cells, in this case of this tumor, can differentiate and become almost normal cells, even though they have the same DNA mutations as the stem cells that are cycling. And this and many other studies from our lab and others have shown that pediatric brain tumors in general, and this one in particular, follow a very similar hierarchy that we know from normal development. Why is this important? Again, we want to get to that next stage, which is to find new treatments for our patients. And knowing now that these tumor cells, at least a few of them, already know how to mature and exit the cell cycle and stop proliferating before treating, can we find and design new treatments that push all of the cells towards more normal cells? And would that be a new way that we would call differentiation therapy to help our patients? The next point I want to touch upon is not only heterogeneity, but also a new appreciation for how tumor cells interact with their microenvironment and also the immune cells. Once we had all these single cell sequenced within multiple pediatric brain tumors, we started to cluster them, and as I said before, found that there were multiple different tumor cell kinds within the same tumor, and also normal cells. And what we found more recently, and also other groups across the planet, is that these different tumor cells and normal cells talk to each other. Work from Michel Monge and others has shown this beautiful interaction of normal neurons within the brain to stimulate tumor cell growth. And we've also learned that the immune cells here in blue go into the tumors in pediatric brain tumors, but don't do quite a good enough job to really kill tumor cells and help our patients. So from this knowledge, we can now again take the next step and design new treatments, first of all, to block communication when it helps the tumor, and such trials are already underway, and or to stimulate this communication in the case of the immune cells that we want to make stronger in the fight against tumor cells. Next, I want to touch upon a new class of drugs that are being developed as we speak, called epigenetic drugs targeting the epigenome. What is the epigenome? It's a way of wrapping up DNA. So the string of genetic information in each cell or DNA is really long, and in order to be neatly wrapped and be organized in a neat way, it's wrapped about those histones shown here as spheres. You can easily imagine that this tight wrapping or loose wrapping is really important because only the open genes, so to say, are the ones that can play a role in the cells. And you can also easily imagine that if this is all messed up, and the control of tight regulation and wrapping is messed up, then all of a sudden hundreds of genes can be active that shouldn't be. And this is exactly what pediatric brain tumor cells have hijacked, and many of our tumors use this mechanism of the epigenome to activate many, many more genes in their cell nucleus than they should be. On the other hand, we've already started to learn how this epigenome is regulated, and not just learn about it, but also target it. And I'm showing just a few examples of drugs that are already partly in clinical trials for pediatric brain tumors, and more of them are about to come, where we then change or try to change the wrapping back to what it should be like. And then lastly, I want to touch upon new disease models. Our field really had a big problem up to 10 years ago, where we just didn't have the right models that would be similar to what we see in our patient tissue. And one of the things we've learned, we and others in the field, is that brain tumor cells just really like being around normal brain cells, as I've shown you before. It's really difficult to reproduce that in the lab, but one way to do that is to grow brain organoids, shown as this darker sphere here, where we grow mini brains, so to say, from reprogrammed iPSC cells, and then bring brain tumor cells in the shiny sphere in close proximity. And what they do then, now the tumor cells are in green, they grow into this mini brain in the laboratory and infiltrate it as if they would in a normal brain. And by doing that, all of a sudden, they start recapitulating, again, all these programs that we see from real patient tissues. I'm just showing here over on the right is a case of a DRBG that is now reproducing the shapes and different states that we had seen by single-cell RNA sequencing. Why, again, is this important? Because this allows us to test our therapies in a more close to normal background before we can go into clinical trials. And all the things that I've shown you and many more are really all at our fingertips now, as technology has changed so much. And we're now ready to take this next step, integrate all this different new knowledge from our group and all the ones around the planet, and hopefully come up with better treatment strategies for our patients. And at the very end, I just want to thank my fantastic lab. We took this picture yesterday for Go Gold in September, Childhood Cancer Awareness Month. And I want to thank all my wonderful collaborators, all of our supporters and funders, including the National Brain Tumor Society. And most of all, a big thank you to all of our patients and families. We could not do this without you, and we really need your partnership in combating this disease. So thank you very much for all you do.
What a great overview and presentation. Really exciting to see all the tools that are being assembled and really understanding, with the cutting edge research that you've done with the single cell sequencing, figuring out how to integrate that. And then, I'm so excited to figure out, talk more about the differentiation therapy and then the organoids. It's really fantastic. I'll hand it back over to Rachael here in a few minutes for questions from our brain tumor community. But first wanted to quickly outline some ways that the National Brain Tumor Society works with pediatric brain tumor community to really make a difference. And this really kind of spans from working with volunteers, donors, government, even all the way to biopharma and other partners to really try to achieve breakthroughs and create change with the goal of really, having a profound improvements in pediatric brain tumor patients. So just a few examples of this are NBTS's Defeat Pediatric Brain Tumor Research Collaborative, which has now evolved to Defeat Brain Tumors, and it's a collaborative team effort. NBTS and its volunteer advocates have also helped influence the federal government to fully fund the Childhood Cancer STAR Act for the third consecutive year, and to fully fund the Childhood Cancer Data Initiative for the second year. Annually, NBTS spearheads efforts to really get the right amount of funding from the NCI, from the Department of Defense, and this is a really big component for our pediatric brain tumor community. And then, some of the things that Dr. Filbin was talking about was, NBTS has advocated successfully with the NCI to extend funding for Project HOPE, which is the high-grade glioma omics in pediatric and AYA populations. This is a collaborative intersecting research project that uses single-cell RNA sequencing, much like what was shown to you in Dr. Filbin's presentation, to learn more about tumor heterogeneity, growth, and evolution. So these are just a few examples of the kind of things that we're trying to make a difference. And you can kind of access all of this in our Childhood Cancer Awareness Month webpage at braintumor.org/CCAM. So with that, let me turn it back over to Rachael for questions from our patients and caregivers.
Thank you so much, Kirk. And also, Dr. Filbin, thank you so much for such a great presentation. Part of what's so great about Informed in 30 is that this is really meant to be an informative piece for our patients and caregivers. And as such, we actually have some questions that we've gathered from our community. So from that sort of patient and caregiver perspective for you, and certainly Kirk, and some of these questions, please feel free to chime in. So to get us started, the new classifications, which actually you mentioned in your presentation, how do they affect current patients in terms of the diagnosis and those that are undergoing treatment?
Yeah, thank you so much again, and also for this great question. You are right that the classification is changing, and there's a new book from the WHO with the new official classification coming out in the next few months. And in terms of what's happening today, there is one trial, for example, for medulloblastoma, where we know of 4 different groups. And there's now one group called Group 3, where a particular addition of a chemotherapeutic agent seems to be more beneficial than for the other groups. So that's just one example where this classification is already in clinical practice nowadays. But I think that we will see many more of those clinical trials come up, and dividing up our patients even more, which is great on the one hand, because it will be more personalized. It brings with it a challenge, of course, because we as physicians will have to collaborate even more, and are already doing that, to get enough patients on our clinical trials that are for very specific subgroups, to get them done in a reasonable time to help enough patients.
Yeah, I guess the one thing I'll add is that, having the new WHO guidelines is it really does help subdivide things, but it's really important that everybody understands these classifications. And so to that end, one of the things that we're doing at the National Brain Tumor Society is that we have research roundtable programs that we come together with the FDA and neuro-oncologists, neurosurgeons, and pharma that are all involved, all the stakeholders. And so we have a December meeting where this is the exact topic, which will try to really help both the FDA understand, but also the pharma companies, and working with the neuro-oncologists and the patients. So just a quick thing there.
Yeah, thank you very much for that. Exciting times, I think, even though I also understand it's frustrating, or it can be frustrating for many families who are being treated today, because the classification is out, but yet there are no or few specific treatments. For sure. But we're working all very, very hard to change this quickly.
Such a great point to make. And as a matter of fact, what you've talked about in terms of clinical trials leads to our next question, which is, what would you suggest to a family that is trying to find a clinical trial for their child?
Yeah, that's also a great question. Of course, there is a website from the government called clinicaltrials.gov, where most trials will be on, because we are required, if there is funding from the government involved, to put clinical trials on. And also most of the industry-sponsored trials will be there. However, it's a not easy to navigate website. So in addition, and it's not complete. So in addition to that, which is a great resource, I think finding your treating physician and really talking to them about, what kind of tumor do I really have? What tests have you done to classify it? To what degree? And then just having a very frank conversation about this very topic. Do you know if there is a clinical trial specific for this class of tumor, subclass of tumor I have? We all know each other in the field, which is one beautiful thing in our community. We all know centers, like who the other centers are, the physicians there, what trials they have open. And we would never hold back sending a patient over to whatever is the best trial for that particular child.
Really great points. And I guess what I'll add there as well is that I totally agree, is that you talk to your treating physicians and really understand, because they are linked up in their network and they know the landscape. In terms of trying to navigate yourself, there is something that also that the National Brain Tumor Society has, which is a clinical trial finder, which has some components that make it easier to navigate than clinicaltrials.gov. And you can find that on our website.
I'll just, actually add on to what Kirk is saying in terms of clinical trial finder. National Brain Tumor Society does have a clinical trial finder that pulls directly from clinicaltrials.gov. And it was actually designed by a brain tumor patient, to make that interface easier and more user-friendly for average folks that want to try and find clinical trials. So that's a great point to make, Kirk. One of the comments that you just made, Dr. Filbin, in terms of biomarker testing, should a family pursue having biomarker testing done on their child's tumor?
Yeah, that's also a great question. I think it depends a little bit of what kind of tumor it is. Not every tumor needs all the different molecular diagnostic tests. But according to the new WHO classification that will come out next month or so, I think that can be used as a very nice guideline of what is the minimum of tests we should be doing for each of the different tumors. And all the pathologists will have it, all the oncologists will have it, and this will really guide our diagnostic classification. In certain cases, however, in the complicated cases where it's not clear, cases in which the tumor comes back and might have, acquired more mutations, for example, then oftentimes it's very helpful to do deeper testing to find, is there any clinical trial that would match? And not every clinical trial, I should also say, will have the requirement of molecular testing before you can get on the trial, but more and more have it nowadays.
When you have someone who has a child who is newly diagnosed, and as we are so familiar, so often, families are thrown into a situation that they're just not prepared for. It's it's something that is surprising, and they're in an entirely new world. If you were working with a newly diagnosed family, could you offer just a couple of key questions that you feel would be most important for them to ask at that critical time, which is the newly diagnosed?
Yeah, I agree with you. This is one of the toughest time, I think, in any person's life, because nobody is prepared. And even as physicians, we have done this so oftentimes it's each journey is different and it's tragic each single time. And it doesn't get better by doing it longer, I should say. So what can you ask in this moment? One of the questions I think I would ask is, have you seen this tumor before? Have you treated this tumor before? Is this place where we're at, is this a center for treating this tumor? I think that's important because some tumors are more common and can be maybe treated in more centers. However, a few tumor diagnoses are so rare that it would be, I think, a good idea to at least get a second or third opinion from neurosurgeons or radiation oncologists or oncologists who have treated this tumor before. And they would also then be able to tell you more about the journeys they have seen before, what they know about the prognosis and side effects and so on. So that's what I would recommend.
Yeah, and I couldn't agree more, is that you can't be shy. You have to really figure out, you go out there and ask the questions. Don't be shy. Make sure that you're getting, the quality of input, the same kind of level that you can get from Dr. Filbin at Dana-Farber or, other centers of excellence. It's really important, and don't back off of that.
Yes, and I want to add that I think social media have been helpful in the last few years to decades to allowing families to find each other, especially with rare diseases. Sometimes, of course, like everything with social media, you have to be careful which group you associate yourself with in order to get the right information. However, I think it's also a big chance to connect with other families who have undergone the same thing.
That connection piece is so important for families, and sometimes sharing that information that can help them because somebody else has already been through it. As sort of a follow-up to that question, so asking those key questions, when you're newly diagnosed, and again, we've identified that as such a critical time and also such a difficult time. Recurrence, kind of the same question. What are some important questions that they should be considering asking when their child's tumor is in recurrence?
Yeah, I think especially, like I said before, at recurrence, that's the point where you want to get multiple opinions from different centers. Because at the beginning, when a patient is newly diagnosed, most of us follow very specific guidelines for the first round of treatment. At recurrence, though, there is oftentimes more options and less at the same time, more options in terms of clinical trials, but you have to find the right one that's for you and your child. So at that case in particular, reach out to different centers, find out what's going on. And I should also say at this point, there are several clinical trial consortia that we are all part of. And we have recently started to work also with patient advocates and families on patient navigators. So hopefully we will have good concepts in the near future where new families can then also reach out to such patient navigators and find out who is doing what, where, what's available, what slots are open, because there's a lot of logistics then involved if you want to get on a new clinical trial.
So, I know that we're starting to come close to our time, but one of the other questions that we did receive from the community, and although I think that some of it has been addressed, I'm going to ask it the way it was put to us, which is that, again, at that sort of critical time of newly diagnosed, what should a family look for when they're looking at a clinician or an institution to treat their child?
Yeah, I think, and this might sound funny out of my mouth, but I think gut feeling is a good one to go after. You have to be comfortable with your physician and caring team. It's more than one physician when you have a brain tumor. You have a whole team out of a neurosurgeon, oftentimes radiation oncologist, psychologists, and many, many others. So I think that the most important is to feel comfortable with them, because that will allow you to ask all these questions. And I think most of us, if not all of us, are really, really happy to address questions that are asked, any questions really, and then connect patients and families to the right place, even if you're not, you currently don't have that trial open at your space. So I think trust and comfort with your primary care team is very, very important. And then asking all these questions and being able to reach out with your team of doctors to all the opportunities that are there for you and your child.
I think that is so helpful to hear that, especially for those that are newly diagnosed. I think that's such a helpful piece of information for them. Dr. Filbin, thank you for joining us today, for creating meaningful content for our community, and for answering their question that helps them to better understand the advances being made in pediatric brain tumor research and treatment, to help provide better outcomes for our patients. And Kirk, thank you as always for helping us to break down these complex concepts for our audience.
Well, of course. I mean, I think the recurring theme here for these informed in 30s, I feel like they should be informed in 60 minutes. Because I always, at the end of them, there's more I want to dive into. So we would hope that, maybe there could be a return performance, Dr. Filbin, somewhere down the road, and we can kind of see where we are a year from now and what the advances. It's been exciting this last year, and I think it's just going to keep building. And so I know there's a long way to go, and we're all committed to figuring that out. But I do believe we're getting close. So anyway, thank you again for everything you're doing and for participating today.
Thank you very much also from my side for today and for everything else you do. This takes more than a village. It takes all of us to work together, and you're setting a wonderful example. So thank you very much.
I also want to take a moment to thank our community for joining us to be informed about innovations in pediatric brain tumors and what that means for patients and caregivers. I invite you to join us in October for our next installment of Informed in 30, Innovations in Brain Tumor Imaging, with Dr. Ben Ellingson from the David Geffen School of Medicine at UCLA. Thank you so much for joining us.